Hi class, in this recording we are going to be focusing on the primary accessory organs of the GI tract or the gastrointestinal tract. And these primary accessory organs are the liver, gallbladder, and pancreas. As our small intestine receives partially digested food, the chyme from the stomach, the small intestine will further process that chyme with secretions from the liver and pancreas. These secretions are going to enter, the secretions I should say from the liver and pancreas, enter the digestive tract in the duodenum. So that first part of the small intestine immediately after the stomach. And they help the stomach, or excuse me, they help the duodenum to process the chyme. One of the key things that we need to look at with our liver here, which is the biggest of the accessory organs, is that our liver is, well, quite large. It's huge. It's the body's single largest gland. A human liver, adult human liver, weighs about 3 pounds, and it's going to be immediately inferior to the diaphragm. So our diaphragm is going to sit directly above the liver, and we're going to have the liver sit on the right side of the body. So it's at the right superior margin of the abdominal pelvic cavity. Now, from a digestive perspective, our liver is going to secrete bile. Or another way to think of the liver is that the liver makes the bile, and then that bile is going to be sent to the gallbladder. And that gallbladder is shown as a green organ right here, although it's not really bright green in real life, just like veins and arteries aren't bright red and bright blue. But the gallbladder right here is going to be inferior to the liver, and it's going to adhere to a depression on the bottom of the liver between the right and quadrate lobes. And as we look at our liver, the liver cells that make up the majority of the liver are called hepatocytes. Oops, I went too far there. Here we go. So hepato meaning liver, cyte meaning cell. So after we eat a meal, hepatocytes from the blood, or hepatocytes within the liver, will absorb nutrients from the blood, particularly glucose, amino acids, and some other miscellaneous nutrients. And these hepatocytes are then going to process them. Sometimes they process them and then re-release them back into the bloodstream to be used by other organs. Sometimes the hepatocytes process them and store them within the liver. Between meals in particular, hepatocytes will take stored glucose, which is stored as glycogen, and break the glycogen down and release that glucose back into the bloodstream. So the liver, and particularly the hepatocytes of the liver, are essential for maintaining blood glucose homeostasis. Our hepatocytes are also able to degrade hormones that we don't want in our body anymore, to break down toxins, and remove bile pigments or drugs from our bloodstream by chemically altering them. Our hepatocytes are also going to release into the bloodstream albumin, which we use to maintain blood osmolarity, lipoproteins, which we use to remove lipids throughout the blood, clotting proteins like fibrinogen, and clotting factors as well, like factor 5, factor 10. They also release angiotensinogen to help regulate the clotting process, and some of the blood pressure hormones as well, so that angiotensinogen can turn into angiotensin, which then is part of the renin-aldosterone feedback system. So the liver does a lot of things. We're going to focus on digestive functions, though, of the liver. And within the liver, there's going to be what is known as a lobule. And these lobules within the liver tend to have a rough hexagonal shape. And this hepatic lobule, or liver lobule, is going to have little tiny channels in it known as bile caniculi. And these bile caniculi are shown cross-sectionally or transversely right here as little tiny green circles. So you might have to get your face really close to the screen to see those. And those bile caniculi all converge at this larger channel at the corner of the hepatic lobule. That larger channel is referred to as a bile ductule. And that bile will converge in the bile ductules ultimately to the right and left hepatic ducts that are still within the liver. And then from the right and left hepatic ducts, exiting the liver, we have the common hepatic duct, which carries bile away from the liver and will send that bile to the gallbladder. And then while the bile is traveling to the gallbladder, it travels through the cystic duct. And then when we need the bile, so we store the bile in the gallbladder and we concentrate it a little bit too. After storing the bile in the gallbladder, eventually we need it. And to release the bile from the gallbladder, we send it through the bile duct down into the duodenum of the small intestine. And that bile duct forms when the cystic duct from the gallbladder and the common hepatic duct from the liver converge and will drain into the duodenum. So here is an actual human gallbladder. And as we look at these gallbladders, I think this is a really cool figure because it really emphasizes the greenish coloration of the gallbladder, though it's not as bright green as your figures may suggest in your textbooks. And the gallbladder, generally speaking, has a pear shape to it, although as I'm looking at this figure, to me it looks more like dog feces than a pear. But it just has a kind of an oblong, rounded shape to it. And it stores and concentrates the bile. So the liver makes the bile, and then the gallbladder will store and concentrate it for later use. To concentrate the bile, the gallbladder removes water from it. The gallbladder is approximately 10 centimeters long, or about 4 inches long, and has a lot of mucosa on the inside to help protect it from the bile it's concentrating. The gallbladder is broken up into 3 regions. We have a head, a body, and a cervix or a neck. And as we look at the head and the cervix of the gallbladder, we have the head right here, and then the cervix of the gallbladder is right here, which would connect directly to the cystic duct. When we think cervix, cervix is a root word that means neck. Most people, when they hear this word cervix, think of the cervix of the uterus, but there's a cervix of the gallbladder. There's a cervix of many different organs in our bodies. Now, as we look at the bile that's produced within our gallbladder, or I should say concentrated within our gallbladder, that bile is a yellow-greenish fluid. It has lots of minerals, lots of salts, lots of acids, and its primary goal is to help us digest lipids. So here is some healthy-looking bile. Notice that greenish coloration of the bile. Within the bile, we have a lot of bilirubin, and that bilirubin is a pigment, the principal pigment of the bile that gives it its color. We get bilirubin from the decomposition of hemoglobin within the spleen. As we release bile into the small intestine, it eventually makes its way to the large intestine, and then the bacteria in the large intestine are going to take the bilirubin and turn it into urobilirubin. That urobilirubin is then going to be further processed, and we'll have stereobilin. That's going to give feces our brown color, and then it also can be turned into urobilin, which I believe is sometimes also referred to as urochrome, but don't quote me on that one. And that urobilin is going to be in the bloodstream and gives our urine its yellow coloration. As we look at those acids within the bile, or all the bile salts, they are going to be a lipid-based salt, or a steroid-based salt synthesized from cholesterol. And these bile acids combine with lectin, which is a phospholipid, and these two work together in order to help us digest and process and absorb fats within the small intestine. If you have a salt concentration in the bile that becomes too high, the salt eventually starts to precipitate out of solution to form gallstones. And those gallstones can clog up the gallbladder. So here we have a gallbladder that's been removed, and that gallbladder had to be removed because it was completely filled up with gallstones. Now, as we look at the bile within the gallbladder, the bile within the gallbladder first gets there when the bile duct overflows into the gallbladder. And that, as we look at the liver, it's constantly secreting bile. It secretes half a liter to a full liter of bile every single day. Most of those bile acids are just going to be absorbed directly within the ileum of the small intestine, so the very last part of the small intestine, and sent back to the liver. And as it's being sent back to the liver through the bloodstream, hepatocytes within the liver will absorb the bile acids and resecrete them into the bile. So we have a recycling system within our liver, but not all of the bile acids that make it into the intestinal tract are reabsorbed. So we have 80% that are reabsorbed. That means 20% is going to be excreted into the feces. Now, as we look at this enterohepatic circulation, this is a circulatory route that goes from the liver to the intestines back to the liver. So we can take a bile acid and we can recirculate it a couple times, 2 or more times, as we're digesting a typical average meal. And as we look at these 20% of the bile acids that we excrete with our feces, this is our body's only way of getting rid of excess cholesterol. So if we have too much cholesterol in our body, we take that cholesterol, turn it into some bile salts, and then we defecate out those bile salts. Our liver is constantly making new bile acids from the cholesterol that we eat or produce, so we're always able to replace the stuff that we've lost through defecation. As we look at the pancreas, the last accessory organ that we're going to be talking about in this video recording, our pancreas is located directly inferior to the kidney, directly pressing against that greater curvature of the stomach. The pancreas is retroperitoneal, which means that it's behind the peritoneal membrane, or behind the abdominal, or posterior to the abdominal pelvic cavity. And the pancreas is a twofer, kind of like the liver, in that it's multifunctional. It's an endocrine organ and an exocrine organ. We've already talked about the endocrine functions in detail in BIOL and AMP1 when we talked about insulin and glucagon and the pancreatic islets. So we're going to focus on the exocrine portions in this digestive system discussion. Most of the pancreas is going to make digestive secretions, the vast majority of the pancreas. And it secretes about 1.2 to 1.5 liters of pancreatic juice every day. What is pancreatic juice? Pancreatic juice is a chemical cocktail of many different digestive enzymes. The cells within the pancreas are the acini cells, and these acini cells are going to release their secretions into small tubes. I'm going to skip ahead a little bit here. They will release their secretions into small tubes, and those small tubes all converge to a larger tube, and that larger tube will then be able to drain the pancreatic juice directly into the duodenum. Let me back up again. So as we look at that larger tube that drains directly into the duodenum, that's referred to as the main pancreatic duct. And the pancreatic duct is going to run lengthwise or longitudinally right through the middle of the pancreas and join with the bile duct at the opening into the small intestine to form what is known as the hepatopancreatic ampulla. And at the hepatopancreatic ampulla, hepato meaning liver, pancreatic meaning pancreas, we have the hepatopancreatic sphincter. So a ring of muscle that controls secretions from the liver and the pancreas as they enter into the duodenum. Now, as we look at the pancreatic juice, that chemical cocktail, it is alkaline, and that's very important because we have lots of acid coming from the stomach and being drained into the small intestine, and we need to neutralize that acid that's being drained in with the alkaline pancreatic juice. The pancreatic juice has a lot of sodium bicarbonate in it, which is the primary alkaline component to neutralize the stomach acid. It also is going to contain other electrolytes and some more water to help digest or dilute the stomach acid. The ducts of the pancreas are going to secrete bicarbonate buffers, which are a key component of the sodium bicarbonate, and will help to neutralize that hydrochloric acid that arises from the stomach. Within the pancreas, we make lots of different enzymes. Some highlights include pancreatic amylase, which digests starch, pancreatic lipase, which digests lipids, and to review, if a name has an ASE as a suffix, that is an enzyme. So ribonuclease breaks down ribonucleic acids. Deoxyribonuclease breaks down DNA, or deoxyribonucleic acids. Our pancreas is also going to release some zymogens, some inactive enzymes. Trypsinogen will be turned into trypsin, and then trypsin within the small intestine digests proteins down into individual monomers. And as we look at the pancreas, there are 3 things that stimulate our pancreas in order to make its secretions. We are going to find that acetylcholine, from the vagus nerve and enteric nerves upregulate the acini cells. And as those acini cells are upregulated, they have more enzymes they secrete, particularly during the cephalic phase of gastric control. So when we're thinking about eating food, we start to make digestive enzymes. Just thinking about that delicious pie causes our pancreas to upregulate its secretions. But these secretions, these enzymes, are going to remain in the acini and ducts of the pancreas until the chyme, or until the food gets to the small intestine, the duodenum of the small intestine. We also find that CCK, cholecytokinin, which is secreted by the duodenum, is going, in response to fats arriving in the duodenum, stimulates the pancreatic acini to secrete their enzymes. And it's also going to stimulate contractions of the gallbladder and the relaxation of the hepatopancreatic sphincter. And this is good because if fats are arriving in the small intestine, we really want to get some bile from the gallbladder into the small intestine to help us digest and emulsify those fats. So we relax the hepatopancreatic sphincter and discharge a lot of bile into the duodenum of our small intestine. Secretin also has an effect on the pancreas. Secretin is going to be released from the duodenum of the small intestine and is going to stimulate the liver and the pancreas to make more sodium bicarbonate. So in response to acids entering into the duodenum, the liver and pancreas are going to secrete more bases into the duodenum to neutralize the acids and raise the overall pH to the correct pH for pancreatic and intestinal enzymes to be activated. This raising of the pH in the pancreas, or in the duodenum, also deactivates the stomach's digestive enzymes that needed the highly acidic environment of the stomach in order to regulate, or in order to be active and digest their respective products. So we're done with the accessory organs of the GI tract. If you have any questions or comments, please feel free to let me know by posting them in the class discussion board or shooting me an email. And as always, happy studies.